Hi guys, this is a Lab 10 review, microanatomy of the oral cavity. It's a super long lab, so we're just going to do our best. So what we're looking at here is the mucosa on the tongue, and then our three different major salivary glands. Then we'll look at the lips, and then we're going to look at the teeth as well. So it's an enormous amount of content. We're doing our best, like always, so we'll just get through it. Functions, mechanical, fragmentation, propulsion, chemical, digestions of foodstuffs we're familiar with what the mouth does, so we'll just get to it. New things are divisions of the oral cavity. We have the vestibule region, which you can see is between the lips, cheek, and teeth, and then we have the oral cavity proper right here. We're going to start off by looking at the lip overall, and let's just get to it. So that said, we do have the oral cavities lined by a special mucosa called the oral mucosa. There's three different types. We're going to look at these two during this lab, but we'll mention masticatory mucosa. The general components of all of our oral mucosa is a moist epithelium with three layers: the stratum superficiale, stratum spinosum, and then stratum basal. Then we have our cells inside of these three regions, and this epithelium include keratinocytes, Langerhans cells, which are macrophages, Merkel cells for touch stimuli, and then melanocytes for melanin. And then beneath that, we have our lamina propria, and we have these ridges coming down from the stratum basal, or excuse me, from the stratum spinosum, lined by the stratum basal, and these are the rete ridges overall. All right, but our three types of oral mucosa: we have line mucosa, we have the masticatory mucosa, which is on our hard palate, on the roof of your mouth, and then we have our specialized, aka tongue mucosa, that we'll be talking about. All right, so our masticatory mucosa, it's found on the hard palate and the gums, right? So you can see it here in yellow. It is keratinized, and in some areas parakeratinized, stratified squamous epithelium. So parakeratinized is where you actually do have condensed, you have some cells with nuclei on the very top, and then we know keratinized, of course, anuclear cells on the top. All right, and then you have your lamina propria, which is loose connective tissue underlying, of course, the epithelium, and the two of those make up our mucosa. So our lip, we have three regions overall. We have the outer cutaneous region, then we have our vermilion red border, which you're familiar with. It's why your lips look pink. This is a transition zone. And then on the inside, we have our first of our three oral mucosas, the lining mucosa. So the outer cutaneous surface is quite literally just thin skin. It's stratified squamous keratinized epithelium with hairs and sweat glands. The vermilion border, as we said, is a transition zone. It's keratinized epithelium with finger-like connective tissue projections that bring capillaries near the surface, which is why lips look red. Then we have our inner lining mucosa, which is stratified squamous non-keratinized and lamina propria, and then it will have minor salivary glands, aka the labial salivary glands, with it, as well as the orbicularis oris muscle, which is responsible for being able to move your lips. Okay, so we'll get into all three of those, starting with the outer cutaneous zone. Again, this is just thin skin, so our epidermis, if you were to zoom in here, is just stratified squamous keratinized epithelium, and then we know since it's a skin, we have our epidermis, and then deep to that we have our dermis, which is made up of papillary and reticular layers. And so we have our epidermis. Deep to that we have our papillary layer, which is loose connective tissue underlying the epithelium, and then deep to that we have a reticular layer, which is a layer of dense irregular connective tissues with large eosinophilic collagen fibers. So we start to see that down here, and you can see that the hair follicles start in the dermis. So here we have our collagen fibers. This whole region right here is dermis, and then past that we would get to our subcutaneous fat. Okay, but importantly, we have hair follicles present, and then we have sebaceous glands draining onto the hair follicles, and then we have eccrine sweat glands, and then apocrine sweat glands, which will also drain onto the sweat. Into the hair follicles, but we'll go over those later on at the end of this lab review section. So then we have the vermilion red border, which is, as we said, this portion of the lips. As we said, it's a transition zone. The epithelium is stratified squamous keratinized, which you can see here. The lamina propria is the loose connective tissue underneath, and you can see that we have these finger-like projections coming up from the lamina propria, and these are the stromal papillae. Okay, you can see those here, not quite as well, and we get some weird cross sections. But the idea is that you have these red blood cells in them, and you can kind of see them, and they're supposed to bring these capillaries to the surface, and it gives the red appearance of the vermilion red border. Okay, then we have our inner mucosal surface, which we see here, and this is lining mucosa. So the mucosa made up of our epithelium and lamina propria is a stratified, as you can see here, stratified squamous non-keratinized epithelium. It's a lot thicker than the vermilion border overall. Maybe it's easier to see that in comparison there. And the lamina propria, loose connective tissue, relatively high cellular density and fine collagen fibers. The submucosa has our labial salivatory glands, which you can see here. These are labial salivatory glands, and they're compound tubulo-alveolar exocrine glands, which you can see here, and it's mixed serous and mucus glands. Deep to that, so we have our mucosa, then we have our submucosa, and the submucosa includes these glands, and then it bumps up to our skeletal muscle, which you can see here. This is a cross section of it. That skeletal muscle is the orbicularis oris muscle, and this is just a creepy picture, so I might be fuel. Okay, not too bad overall, but moist stratified squamous, and then you have the labial glands underneath, and then beneath that you have the orbicularis oris muscle. All right, so then moving on, we are going to take a look at the tongue. This cross section of the tongue is flipped. So here we have the dorsal, aka top border, and then we have the bottom ventral surface of the tongue on the top here. And then the tip of the tongue, this set of slides was stained with trichome dye, so collagen is blue, the muscle is red. The tongue has three layers of muscle, which we'll get into to allow for flexibility in speech. And then as we'll see here, we have minor salivary glands, aka the lingual glands. They're mixed mucus serous. It has all of those, and then along our surface of the top of the tongue, it's our specialized oral mucosa. It has four different types of projections, these lingual papillae, which again we'll talk about. And then on the bottom of the tongue, we have, this is just lining mucosa, so same as the inside of the lip. Lining mucosa, stratified, squamous, non-keratinized epithelium. Okay, and moving on. So what are the muscle layers? Well, so this is a cross-section from approximately right here, but there's 3 layers of muscle that are perpendicular to each other. You can see on this is the ventral bottom surface, this is the dorsal top. You have longitudinal muscles going here, longitudinal muscles going here. You have vertical muscle moving down like this, and then you have horizontal muscle in between those. So the arrangement allows for precise movements during speech. All right, so we said that the bottom surface is special, or is lining mucosa. So stratified non-keratinized epithelium, you can see that here. And then deep to that, we have our collagen, our lamina propria, and importantly, there's no submucosa on the tongue at all. You go directly from lamina propria to muscle, okay? No submucosa tongue, no submucosa tongue, okay? And so the consequence of that is the minor salivary glands that we'll see here are directly in the muscle. They're not in the submucosa. Okay, then on the dorsal side, we have the specialized oral mucosa. It's stratified squamous keratinized, and it does have lamina propria underneath. A very thin layer, right? You can see it here, this blue collagen layer. But stratified squamous keratinized versus the bottom, which is stratified non-keratinized squamous. Okay, so keratinized on the roof of your tongue kind of makes sense, right? Because that's experiencing a lot of friction. And then on the bottom, you have non-keratinized. Okay, so let's get to it. We did mention these glands, which are in the back of the tongue right here. So these are the minor lingual salivary glands. So lingual salivary glands, again, no mucosa, so directly in the muscle. And then we can see that there's serous glands right here. Notice they're dark staining. And then these are our mucus glands overall, but they're directly in the muscle. And you can see longitudinal muscle here, you can see vertical muscle and horizontal muscle overall, and again, skeletal muscle. Okay, so then on the dorsal side, the top of the tongue, we have all of these weird projections that go into upwards, and so they have different functions, but primarily they're based, 3 of them do taste perception, and then the most numerous one is just for friction to help move food. Okay, we'll talk about each one of these in turn, but again, just take a look, salivary glands, the labial, or the lingual salivary glands, and then we have the rest of the papillae, the filiform are everywhere, then you have the fungiform, and those are scattered amongst the filiform, and then we have the circumvallate, which there's only a few of those, and then we have folate, which are located along the sides of the tongue, but we don't have to ID those. All right, so filiform and fungiform. Filiform is the most numerous. They look like this. Right, you can see them here. So you see that keratinized epithelium, and then you see that it has connective tissue below it with its core of lamina propria connective tissue. The function is to increase friction. You can see the electron microscope version of them right here. Fungiform are scattered along the interior 2/3 of the tongue. They're concentrated on the tip and lateral surface. You can see, I suppose maybe it looks a little bit like a mushroom, so fungiform. And the epithelium is also keratinized, and then it has a connective tissue core, and it does have taste buds. We can't really see them very well, but you have taste buds along the upper surface of papillae only. Here'd be a taste bud right there. Okay, but filiform, fungiform looks like a mushroom with little projections upwards, as it has these taste buds embedded on the top surface. All right, then we have our circumvallate papillae, and there's not very many of these, only 10 to 12 along the V boundary called the sulcus terminus. So these are the circumvallate papillae. They're associated with a gland called Ebner's gland, and we'll talk about that in a second. But these 10 to 12 total in the tongue, they're on that V-shape boundary, and they have keratinized epithelium, which you can see right here, keratinized squamous. They have a moat around them, and along the edge of the moat, you have taste buds, which you can see here. And then at the very bottom, you have minor serous salivary glands called von Ebner, and those are located right here. You can see that they're darkly staining and therefore serous. The function of these papillae are for taste perception, and you can see that its lamina propria is vascularized loose connective tissue, right? And then this is a electron microscope version, and then notice you have a moat around it. So you think moat and you think circumvallate. There is a moat around the circumference of the vallate papilla. Okay, and so just one quick thing: the von Ebner works to clear the moat, and it has lingual white paste that it excretes as well. Okay, our taste buds are oval groups of sensory cells. We said that they're in all of the papillae except for filiform, and they are primarily in the, again, epithelium of the papillae. There's 3 types of cells in the taste buds, so you can see taste buds. We have our sensory cells, which have microvilli, taste hairs. They synapse on a afferent nerve, and then there are supporting cells located here as well. So supporting cells we can see here, and then sensory cells. In particular, we were told we wouldn't have to be able to tell the difference between these two because you can see they are hard to tell apart, and they both have apical microvilli. And then we have basal cells at the bottom, which you can see here, and they undergo rapid renewal and replace the other cell types every 10 to 12 days. Importantly, the sensory cells themselves have different receptors. So we have the ability to salty, sour, sweet, bitter, and umami based on sodium channels, hydrogen ion channels, glucose G-coupled receptors, quinine for bitter, G-coupled protein receptors, and then monosodium glutamate for umami for G-coupled protein receptors. Okay, so then we are moving on to the tooth. There's 2 parts of the tooth. There's the crown, which we can see here, and then there's the root. Components of the tooth, we have the pulp cavity right here, there's the dentin right here, there's the enamel on the crown, and then lining the edge of the tooth is the cementum, and then we have alveolar bone adjacent to it, and we'll talk about this periodontal membrane in between a little bit as well. Okay, so this is the cross-section given to us in lab. Not too bad. We'll start with dentin, which is located here, all around the pulp cavity. It's a whitish material with many dark lines radiating outwards. There's one opening called the apical foramen, and it's yellow, semi-transparent. It's about 70% inorganic, which means it's harder than bone. Most of the organic material consists of type 1 collagen fibers, and it's produced by these cells called odontoblasts, which lay down pre-dentin, which then mineralizes into dentin. You have these cells, which are lined on the inside of the outer edge of the pulp inside layer of the dentin, and you can see their nuclei are on this inside edge, and they extend processes into dental tubules, which are very strange looking, and you can see those go to the outer edge of the tooth. And so they can help with, sensing of hot, cold, acid, pressure, etc. Okay, so dental tubules, those are these white spaces right here. And then, the cells retreat as predentin is laid, which means that as you get older, the pulp cavity gets smaller and smaller overall. Okay, and moving on. All right, then we have enamel on the top of the tooth. It is acellular. It's the hardest substance in the body, 96-98% organic. It's composed of prisms or rods, which you can see here, of calcium hydroxyapatite phosphate crystalates. You can also see that here in electron microscopy. There's organic material that is around it, and this is enamelin, and you can start to see that here. The enamel is produced by cells called amyoblasts before tooth eruptions. So they would be on the outer surface of the enamel, and then they die off as the tooth erupts. Okay, and so in our histo lab itself, you can see the enamel here, and it's always going to be perpendicular to these lines that you can see, and these lines are called the lines of Retzius. Basically, this tells you how the amyoblasts were aligned at a particular point in time, and then if you were to zoom in, you can see our actual enamel crystals, the calcium hydroxyapatite phosphate crystalates, which are in the shape of these enamel rods that fit together. Okay, so not too bad. Then we have cementum, and it's a thin white layer, which you can see here lining the root of the tooth, and it's similar composition to bone, 50 to 65% inorganic salts, and then it contains type 1 collagen. It's aneural and avascular, and it's secreted by cells called cementoblasts. And then, as we'll see, so we have an acellular cementum, which is adjacent with the enamel, and then on top of our dentin everywhere. But it can be laid down through the life of the cell, and then the growth is appositional only, so it comes outwards along the outer edge of the cementum. So we have our cells here, and they can get trapped, and they'll get trapped and become cementocytes, and then you have these cells on the outer surface in both the acellular region and then the cellular region. But the cellular region, which we can see here, you have lacunae present, and then canal iculi radiate from the lacunae, which you can see here. There's no Haversian canals, and then a living tooth, the cementoblasts would be on the surface here, but we don't see them because this is a slide. Okay, then we have our pulp, and just notice loose connective tissue. You do have fibroblasts theoretically here. It's really small. Our odontal blast layer, which is producing predentin, so a layer of odontoblasts, and then here you have dentin, and then this is the predentin that has yet to mineralize, that's produced by the odontoblasts. Okay, and then remember, cementum is produced by cementoblasts, and then enamel is produced by amyoblasts, right, which kind of looks like the word enamel a little bit if you were to mix it up. Okay, then we have gingiva, aka the gums, and it's masticatory mucosa. We said it on the roof of your mouth, and then the gums, you have this masticatory mucosa. And so this is stratified squamous keratinized epithelium. Makes sense because it's going to be a friction surface, so it should be keratinized, and then it has a lamina propria loose connective tissue underneath, which you can see here. Functions is to form a junction between the enamel of the tooth and this epithelium via hemidesmosomes, and then prevent foreign materials from getting into the root, unless apparently you are a piece of popcorn. Okay, so not too bad. Deep to that, we have the periodontal membrane or ligament right here, and so basically you have bone that the tooth is embedded in, and so this periodontal ligament is just connecting the bone to the cementum, and it's just an attachment suspensory ligament, and it does contain blood vessels, lymphatics, nerves, and then Sharpie's fibers are present to, which is type 1 collagen fibers that are embedded in the cementum or the bone, and they help basically attach the periodontal membrane, this ligament between the bone and the cementum slash dentin. All right, so then we had a developing tooth. This is already going so long, but we're only like 2/3 way done. You have this is a cross-section of developing tooth. So the crown is developing, the top of the tooth. You have this M-shaped structure, which would be the developing crown. The whitish material is the dental papilla, and it's consisting of mesenchyme. And then this is the future dental pulp overall, again. And then this U-shaped part right here is developing alveolar bone, and you see bone spicules right there. All right, so if we zoom in on this section right here, we can see the enamel and dentin being laid down. So starting right here, we're at the pulp area. This is the mesenchyme of the dental papilla, which will form the future pulp. Then deep to that, we have our odontoblasts, which produce dentin, but specifically they're producing predentin, which will harden into dentin. And then you have our dental enamel junction right here, which means that this is enamel that's been laid down. Remember that our amyoblasts are on the top surface. So here we have connective tissue covering our developing tooth, and then we have our amelloblasts, which are producing the enamel, and here's the enamel that they produced. And then once the tooth erupts, these guys unfortunately pass away, and the outer surface is now our enamel. Okay, so connective tissue underneath that, our tooth is not erupted yet, so we have our amelloblasts, then we have our enamel, then we have our dentin, then we have our predentin, then we have our odontoblasts, and then we have our dental papilla overall. Okay, now we have our glands. So we have 3 major glands, and they're producing saliva. And we like saliva because it helps us break down our foods, but we have the biggest one, which is the parotid glands. Then you have the submandibular and the sublingual glands, and they're all dealing with compound tubular alveolar glands. But specifically, how we're going to look at these is the parotid gland is all serous secretions. The submandibular is a mix. The sublingual is a mix, but the submandibular is mostly serous, and then the sublingual is mostly mucosal secretions making up the saliva from there, right? And then you may remember, rest and digest, we said that from physio, the autonomic nervous system, the sympathetic nervous system, and the parasympathetic will stimulate the production of saliva. And so you may remember that the sympathetic nervous system produces a thicker saliva than the parasympathetic nervous system, which produces a watery saliva. All right, so just to get you thinking maybe. All right, so we have our saliva. We know it's involved in digestion of sugars. It starts with it, lubrication, protection of the mouth, antimicrobial properties. It has a high concentration of potassium and bicarbonate, low concentration of sodium and chlorine. It has alpha amylase to help with the sugar digestion, lingual lipase, which we said came from the von Ebner glands on our tongue. And then we have our mucins, mucus, silomucin and sulfomucin, which serve as a lubricant protective functions, and then we have antimicrobial functions in our saliva. Our components: lysozyme, break down bacterial walls; histatins, antifungals and antimicrobials; cystatins, protease inhibitors, which block the action of microbial proteases, which break down proteins of the human body, which we don't want. And then we have our lactoferrins. They bind and destroy cell membranes and chelates iron necessary for bacterial growth. You have peroxidases and that catalyze production of hypothiocyanate, which is an antibacterial and antifungal agent, and then we have secretory IgA, which is an antibody that neutralizes bacteria and viruses. So basically protection and start of digestion. So we have these three glands, and the architecture for them is all the same. The only real difference is their composition of serous versus mucus cells. So starting with the architecture, we said they were tubulo-alveolar compound, and so you can see that here. Alveolar, they have these bulging out sacs, and then compound, we have branches. And so our secretory cells are arranged into these acini, and they will drain their product into intercalated ducts right here, and then those will drain into striated ducts. And both of these intercalated ducts and our striated ducts will modify the secretion to produce them, to make the secretions into saliva. Okay, but this defines a secretory unit, and then we have multiple secretory units together. But importantly, these units are made up of these acini, which can be contain serous or mucus acini, and then we also have these myoepithelial cells, which are contractile, that help to empty the acini into the intercalated ducts. Okay, so what are these cells? We have the serous acini cells, which you can see here, right? It follows the same pattern as all serous versus mucus. Is it darkly stained? Serous. Is it lightly stained? Mucus. Okay, serous produces water secretion, amylase, lysozyme, statins, cystatins, lactoferrin, peroxidase, secretory antibodies, IgA, and then mucus is producing our mucus overall, so silomucin and sulfomucin. And then down here, we have our myoepithelial cells, very darkly stained. You can see that located here. Here's our acinus with all of the different serous and mucus, but then at the base of that, we have our myoepithelial cell, which is contractile, and it's going to push the cells to release their contents into the intercalated ducts. Okay, so major salivatory glands, the types of acini, well, basically you can just have mixed, mucus only or serous only. And so again, this is pretty straightforward. Here's a serous only one, it's all darkly stained. Here's a mucus only one, and then here's a mixed. And notice how the serous tend to be on the edge, and then here we have our myoepithelial cell overall. Okay, so we've already kind of hinted on this, but we have our secretory unit right here, and then multiple secretory units together so secretory unit, multiple together will form a lobule that's surrounded by connective tissue. Each saliva from each lobule will be drained into a large striated duct. So we have our intercalated ducts that drain into a striated duct, which you can see here. So intercalated, striated. And then after that, you have our then you go to interlobal ducts in between lobes, which are a category of excretory duct. So after our intercalated, then you go to striated, then you go to our excretory ducts, and basically they just get bigger and bigger. So you start with interlobar, then you have lobar, and then you have main duct overall. All right, but importantly, as you get to excretory duct, you're going from simple layered to stratified. All right, so that's the big difference. If you see stratified, you should say excretory duct. Okay, multiple lobules form a lobe. So lobule, multiple of those form a lobe, and then multiple lobes will form the gland overall. All right, so back to our intercalated ducts right here. We just want to talk quickly about what they do to modify the secretion. They contain our friend carbonic anhydrase. They are, as we said, everything below excretory is simple. So this is simple low cuboidal epithelium. And so you can see an example here. And then we have, what are they doing to saliva? They're secreting bicarbonate into the fluid, and they're absorbing chlorine ion out of the fluid. Okay, so the intercalated will go dump their product into the striated duct. And again, this is still interlobar, so within the lobe. Lobule, interlobule. And the striated ducts are going to do a little bit more. Their electron microscopy image will show that you have these basal folds, so basal infoldings right here. Very hard to tell otherwise. Maybe you can kind of see them on the light microscopy, but I don't think we have to actually identify that. Importantly, we have striated ducts are simple columnar epithelium. They're secreting bicarbonate just like the intercalated ducts did, and then they're also secreting potassium into the fluid, and they absorb sodium and chlorine from the fluid, and then aldosterone is going to act at these striated ducts and reabsorb calcium or sodium and help with the secretion of potassium. All right, so these ducts have exchangers to accomplish this. So you have chlorine bicarbonate exchanger to release bicarbonate into the lumen, and then chlorine is reabsorbed from the lumen, and then you have our sodium potassium ATPase, which is allowing sodium to leave and then reabsorbing potassium into the cell, which we know then potassium will eventually go into the duct. Okay, so striated ducts, bicarbonate into the fluid, potassium into the fluid, take out chlorine and sodium from the fluid, and then intercalated ducts, which drain into these guys. The striated ducts are also putting bicarbonate into the fluid. They're also taking chlorine out of the fluid, but they're not doing the potassium and sodium, which is the striated ducts are doing with the help of our friend aldosterone. Okay, so our excretory ducts are these three. So once you get past the lobules, you have their extra lobule, and they're surrounded by connective tissue, which is how you can tell them apart, but they also are pseudo-stratified to stratified columnar. Okay, then we have now we have basics, our parotid gland. The structure of these is, the way that you identify them is serous acini only, and it has long intercalated ducts, which you can see one right here, and the intercalated ducts are simple cuboidal because it's still within the lobe, and you can see it's surrounded by acini, which is how you know it's within the lobule. Keep saying lobule. And then you have our striated ducts, and so here's a cross-section of an intercalated duct. You can see cuboidal overall. And here's another striated duct, and it drains in, or excuse me, intercalated duct, and it drains into a striated duct right here, which you can see is columnar, not the best cross section, and then it has the basal striations, which again, I think you would need to see it in the electron microscopy to be able to tell. And then finally, you have our excretory ducts, which the sign to know that is twofold. One, it's surrounded by connective tissue, and then two, it is pseudostratified or stratified overall. All right, our submandibular gland right here is mixed secretion, but it's mostly serous acini. And so the challenge here is, are you looking at a case of a mucus acini, or are you looking at a piece of fat, fat cell? And so, you know, fat cells look like the signet ring, right? And the mucini are going to be slightly darker staining. But here, the point is, notice how it's mostly serous, and then you have some mucus overall. All right, so submandibular is mostly serous, some mucus. Okay, the ducts are the exact same pattern as for the parotid, meaning intercalated, simple cuboidal, striated, or simple columnar, and then excretory surrounded by connective tissue, and they're going to be stratified columnar or pseudostratified columnar. All right, then we have our sublingual, and the difference here is it's mostly mucinous acini. As you can see here, mostly mucus, and then you have some serous overall. Okay, the interlobar ducts are poorly developed and not as prominent, but you do have mixed acini with serous demilunes. So these are the serous demilunes. So I suppose it looks like a part of a moon demilume. And so serous cells at the bottom, mucus cells at the top. All right, so now we are just about done. Quick review, zones of the lip. We have the outer cutaneous, then we have our red border, vermilion red border, then we have our inner oral mucosal layer. The line mucosa we know goes with the inner layer, so stratified squamous, non-keratinized. Theoretically, it's not undergoing a ton of friction. And then here we have the stratified squamous keratinized with capillaries near the surface, which is our, and they have the stromal papillae coming up, and then thin skin for the outside. All right, just to review thin skin. So we have apricot sweat glands, which are not associated with hair follicle, and you'll notice that they stain very darkly. And then you have apocrine sweat glands, which have big lumens, and they should theoretically be connected to a hair follicle. And then you have sebaceous glands, which are also connected to a hair follicle, and you don't see these large lumens on the inside, but they are very stained, washed out looking. All right, so sebaceous glands, not huge lumens here, so this is eccrine overall. Okay, developing tooth we're familiar with. So here we have our amnioblasts, which are producing enamel. Then we have the enamel dentin border. Here we have dentin. Here we have predentin. Here we have odontoblasts. Here we have the pulp cavity, and then here's connective tissue on top of the tooth. So not too bad. Developing tooth, this is bone. So you have the cells within the bone spicules. So these guys are osteocytes inside of lacunae, and then along the edge you have osteoblasts, and they produce bone matrix. And then this is mesenchyme, loose connective tissue, and this is intramembranous ossification. All right, so then just a little bit more of this. The cells along the edge of the spicule are, these guys, here's what we're talking about. These are osteoclasts to degrade bone, and they're derived from monocytes overall. All right, okay, back to it. Here we have the submandibular glands. Identify the intercalated duct below. All right, so intercalated duct is the first duct, and we can see one here. You have simple cuboidal. They drain to striated duct, which are simple columnar. There's a bunch of them here. Overall, we know that they're larger than the than the intercalated ones, and so we've really only been shown cross longitudinal sections of these guys too. Then we have the excretory duct, and you can see one here, stratified columnar surrounded by connective tissue. All right, here we just have a diagram of salivary glands, so intercalated ducts, striated ducts, okay, and then this is a mixed mucinous, and we can see your serous and your mucinous, serous, and then myoepithelial cells to help get their secretions into the tubules. All right, then we have our intercalated duct junction with the serous acini. Okay, so this is our duct overall, and then serous acini. This is in the parotid gland. Lots of secretory granules, which is to help trigger the fact that it's serous, and yeah, not too much to see here. Capillary. Okay, sublingual gland. We know sublingual is mostly fat, or excuse me, mostly mucus. And so here we have mucus cells on the left, and then we actually have some serous gland cells on the right as part of a or a demilume overall. And then this is a myoepithelial cell. Okay, and there's evidence of some fibroblasts here. All right, that's it. Good job. It was super long, and on to the next one.